right, good afternoon, everyone. It's my honor to present Alex Cormack. She grew up in New York and got her environmental biology degree, bachelor's degree at SUNY, the State University of New York College of Environmental Science and Forestry in Syracuse. After that, she did a senior cruise where she studied the abundance of yellow morph of Phytes asteroides, which led to her, I hope at this point still, love of coral and desire to do coral research, which brought her here. I'd like to applaud Alex for her perseverance in her project. She might elude it in her methods, but she went through a lot of iterations before she got to the actual results that you're going to see today. In addition to that, I'd like to commend you for all the metrics that were thrown at you consistently and very spontaneously. I know that was a lot of work, and this is probably going to be the most comprehensive restoration study once you finally culminate what all of that means. So with that, Alex Cormack.
So I want to talk to you guys today about a little bit of the first part of this big comprehensive project on microfragmentation as a tool for coral restoration. And we want to investigate some of the physiological factors on survivorship of microfrags after they're cut. This is a little time lapse of me actually microfragging. I wish it went that fast. It's a super long process. So we're going to switch slides again. All right, so to start, I want to give a little bit of an introduction on corals and coral reefs, especially for those who don't specialize in this. For context, for those of you who don't live in St. Thomas, that is not a photo from St. Thomas. That's from that's from Australia. But I just want to start with corals are colonial animals. They're made up of tons and tons of tiny little polyps. They have their own tentacles and mouths. Scleractinian corals are what we're going to focus on today. They utilize a process called calcification, and they use that to secrete a calcium carbonate skeleton. Coral reefs are complex structures built up of the accumulation of these calcium carbonate skeletons. I say this to highlight that corals literally build coral reefs, making them some of the most important animals in the ocean. The complex reef structure, right, that's got all of these different mounds and hidden spots, a lot of architectural complexity. It protects our coastlines from erosion. It stores carbon in it, which is a big hot topic when we talk about climate change. And it forms the basis of the most biodiverse ecosystem on the planet. So all this to say, really, really important. So as many of you know, there's lots of threats to reefs right now. Many of us here have seen how climate change is impacting coral reefs. Thermal stress is probably the biggest threat to coral reef ecosystems worldwide. Elevated water temperatures for extended periods of time cause massive stress events in corals. They can be so severe that an event can wipe out over 75% of live coral cover. As these events are increasing in frequency and severity due to climate change, our reefs, especially in Florida and the Caribbean, are shifting from reef building to net eroding. So what this means, 10% live coral cover is required to keep a reef in a state of net building, where the corals are building structure faster than it's eroding away. Net erosion occurs as a combination of factors, including ocean acidification. It leads to corals not being able to build as much as is being eroded away, both by natural processes, by herbivores grazing on the reefs, by just the acids in the water dissolving. The reef loses complexity, becomes flatter, and that can cause the reef to stop providing that coastal protection and habitat. And it also prevents it from keeping up with sea level rise to keep the corals near the sun, which we'll talk about in a second. A big question, all this to say, is that in restoration, of course, we want to know how can we increase coral cover. Restorationists have been fragmenting corals for years now. Corals are colonial. they reproduce asexually and sexually, but asexually, they reproduce through fragmentation. So if you break them apart, they can each grow on their own. All those little polyps keep proliferating and growing out. Microfragmentation specifically as a new technique was started in around 2014 at Moat Marine Lab as a method to speed up the growth of corals. So in this method, they cut corals specifically into 1 centimeter squat size pieces or a little bit bigger than that. By doing that, you can induce a rapid growth. Very small pieces have been found to grow up to 10 times as fast, sheeting out their tissue onto surfaces. These slow growing, we do this on even slow growing massive corals like Orbicilla flavellata that are slow growing and massive. There are kind of primary reef builders, especially here in the Caribbean, Orbicilla flavellata, which we'll talk about a lot today. It's my study species. It is our primary reef builder, so super important for all those things I talked about before. But again, they're slow growing. It's right in the name. So restorationists want to try to find as many ways as possible to get them to grow faster. The benefits of this technique are that you get this fast surface area growth. They sheet out. They can actually, if you put multiple corals of the same genotype next to each other, they'll fuse back together into a larger coral again. The smaller the fragment, kind of the faster the growth, right? And it's relatively quick and easy. It's easy to teach other people to do it. There are some possible trade-offs with this, right? So what we're going to talk about is 42% or lower survival after outplanting. They're very vulnerable, especially to predation from herbivores, and also just general mortality, which is what we're going to be talking about today. There's possibly some growth trade-offs, which will be looked at in the future. There's also just a lot of mortality post-fragmentation. You cut them and they start dying, and then they'll level off, and that's when they're ready to go and you can outplant them, you can use them in restoration and stuff, but not a lot of studies have looked at this death that happens immediately post-fragmentation. So that's what we're going to look at. We want to look at the potential drivers of this mortality post-fragmentation. And what that can tell us is a little bit about why they're dying, and also if there's any ways we could maybe select for different corals and things like that, maybe aid them in survival. We're going to look at the size of microfragment cut. Like I said, the smaller the size, the faster they grow. But there might be a little bit of a trade-off there. We're going to look at their tissue biomass, their proteins, and their carbohydrates in the tissue. And we're going to look at algal symbiont community composition. And that seems a lot right now, but I'll explain them one at a time. So first, while 1 centimeter size squared fragments have the potential for rapid growth, they may be incredibly vulnerable. That 42% comes from a study that looked at frags after they were outplanted. And they found that those 1 centimeter squared frags had a 42% overall survival, while fragments in a 3 to 6 centimeter squared size class fared much better, around 70% survival. Still, this study quickly mentioned that even more fragments died directly post-microfragmentation and weren't included in the study. And we see that happening a lot where it's very quickly mentioned that, yeah, we lost a lot like right after we cut them, but like that always happens, so we'll move on. It could be possible that these smaller sizes are a little too vulnerable, even though they have that faster growth. Another driver is within this cup of goop. It could be the tissue biomass and the energy hosted in the coral tissue, those reserves. We're going to talk about tissue in the form of ash-free dry weights, which is a metric of tissue thickness. And then the protein and carbohydrates, which are just like in us, they're energy storage in the tissues, kind of the reserves that the coral has built up over time. We'll talk more about it in a second, but bleaching events lead to mass death of corals, mostly due to starvation. So having lots of tissue and energy stored is vital for surviving bleaching. So corals that survive bleaching typically have thicker tissue, more proteins, more carbs, more lipids, things like that. So it could be possible that the energy storage and tissue biomass could impact microfragments survival during the mechanical fragmentation stress. We're gonna talk about algal symbionts, but what symbionts, right? So I'll take a step back and explain a little bit. Inside the coral tissue, there are dinoflagellates they're called Symbiodiniaceae. They give the coral that brown color and they photosynthesize to provide energy and carbon. The coral in turn uses that carbon to make skeleton. They make a structure called a calyx, which is an almost perfect structure for light capture, therefore helping the algae capture light, right? That symbiosis, that back and forth. Coral bleaching is a stress response where the coral loses their symbionts, turns white because the symbionts are gone, it's lost that brown color and you're seeing the skeleton. That's why they're so bright white. The coral can recover, but if the temperature doesn't cool down or if the coral doesn't have enough energy storage, it will likely starve to death. They're also more vulnerable to to disease and predation and other things. But so just remember that coral bleaching is a loss of those symbionts. There are 4 genera of symbionts. We've got in yellow, just to color code them from left to right, we've got Symbiodiniaceae. Symbiodinium, that's what that's supposed to say, not Symbiodiniaceae. We've got Symbiodinium, Breviolum, Claudicopium, and Durastinium. So each genera has an impact on various metabolic functions within the coral. They're all a little bit different from one another. Most corals prefer a symbiont and they stick to it, but Orbicilla favellata is a little bit of a generalist and it'll host all 4. So during bleaching, we're going to use an OFAV as an example because that's all we're going to talk about today. That's an Orbicilla favellata. During bleaching, though the coral is white, it still has some symbionts left. Usually those left behind are still performing well, sometimes tolerant to the heat. Those left behind will proliferate as the coral recovers, and thus the symbiont community composition changes. Why am I talking about this if we're not going to talk about bleaching today? I want to give you an introduction to symbiont shuffling. This is when it mostly happens, when that community changes over time. It's less likely that the coral is picking up new symbionts, and more so, there's an internal competition between all the symbionts inside them, like a little ecosystem going back and forth. So we'll go back to this slide. So the different genera have different metabolic functions, right? So maybe that could be a driver of mortality. Maybe some symbiont genera make them more vulnerable or make them more likely to survive fragmentation. What we've seen in some past study is corals shuffling over to Durastinium trenchi in the water tables. So we have this symbiont in the Caribbean that's heat tolerant, but it's opportunistic. So during bleaching, it'll proliferate in corals and dominate their community structure. It's invasive here and it has some trade-offs, like it might lower their growth rates. Nursery corals at moat, when compared to wild conspecifics, were full of Durastinium trenchi, whereas the wild corals were not. So it could be from nursery conditions. It also could be from the restoration techniques, such as microfragmentation, which is why we want to talk about that today. It is unlikely that mechanical stress could cause symbiont shuffling and opportunism like bleaching does, because it doesn't cause a loss of symbionts, but we are going to take a look at it and see if that fragmentation stress and turning them into microfragments will trigger the symbiont shuffling, which is why I wanted to explain all of that. I know it's kind of a lot. We want to look at 3 big categories. We want to compare the mortality post-fragmentation of larger fragments, which are about this big in size, about 63 to 130 centimeters squared, to microfragments from the same corals. So collecting the same corals from the field and cutting different things out of them. What I mean by genet is that the fragments all came from the same coral out in the field, the same genotype, the same coral individual, colony, however you want to call it. I'm going to call it a genet today. We wanted to look at how size and genet are factors influencing this mortality post-microfragmentation. We hypothesized that smaller microfragments would have lower survivorship and that the different coral genets would have different mortality from one another. Some coral genets are maybe better suited to microfragmentation than others. Then we want to look at some reasons of why that might be. So some physiological differences between high and low mortality. We hypothesized that corals with low mortality, so high survivorship, post-fragmentation, have high tissue biomass, proteins, and carbs. That the thinner corals with less reserves are going to have high mortality, low survivorship. We also think that the symbiont community composition will be different in corals with high versus low mortality. We do hypothesize that after some time in the nurseries, the corals will shuffle their symbionts, but that the microfragments will not be different from the large fragments. So how are we going to do this? First step, to look at fragment survivorship, we've got to make some microfragments. So we go out to Perseverance Bay. We collected 15 colonies of Orbicilla favellata that were relatively far away from one another. I tile sawed to cut them into pretty big pieces, so that's where I got those larger fragments. And then I used another saw to cut about 35 microfragments. They're all a little different from one another, but that was kind of the ballpark. And then over 25 days, I tracked their mortality. If it had more than 50% tissue loss, I counted it as a mortality because in past iterations of the experiment, we did leave those corals in the table, and that's not a good idea. So we did 25 days of monitoring. So first up, I want to talk about the size. I did not cut my microfragments 1 centimeter squared in size. In past iterations of the experiment, I did do that. I cut them very, very small, and I lost all of them multiple times over. So this time I went a little bit bigger. They're between 2.7 and 7.8 centimeters squared. So relatively big, but they're still very small, okay? They're still microfragments. There are some size differences between the different genets. This is just looking at the size of the microfragments along each of the 15 genets. And you can see that there's some differences. So I cut them a little bit different, but we do have a wide size range. And I will say that while you can see M here was cut larger than most, and O here was cut smaller than most, those are my two with the highest mortality. So I looked at the relationship between size and survivorship with a binomial general linear model. And I found that there's a slightly positive, but overall not significant relationship between size and mortality in this size class. So if we included those smaller fragments around 1 centimeter squared, we might have seen a significant difference. But in these, the mortality was about the same across all of them. So next, I want to talk about the microfragments versus those larger fragments. This is a Kaplan-Meier plot. There's day-sense microfragmentation, or fragmentation in general, on the bottom, 1 through 25. And then on the side there, we have survivorship, or the proportion of frags alive. So the top is a proportion of 1 or 100% survivorship. So a log rank survivorship test found significant difference between microfragments and large fragments. This is first to show that 100% of those large fragments, I had 15 of them, they all survived. So first of all, I'm not terrible at coral husbandry. Second of all, when I was cutting them, I didn't totally like drop them on the ground and stuff. Third of all, they were still cut with a saw. So it's not the fragmentation itself that's causing mortality, but specifically how small the microfragments are. This also is a really good show of the general trend in survivorship in all of the microfrags in the first 25 days. So we see this big dip from 9 to 11 days where I lost a lot of them, but then they kind of level off, and after 25 days, we stopped losing any, which is a really good thing for restorationists to know. That kind of need about 2 weeks, and then the ones that are left behind still alive are probably going to keep surviving. As long as you remember to clean them and feed them and baby the heck out of them all the time. So next I looked at just the microfragments separated by genet with their survivorship. It's kind of a lot. Let me break it down for you. Same as the last plot. This time each line is a different genet. Same log rank survivorship curve found a significant impact of genet on mortality. So we see there's kind of this large gap around 50% survivorship of those that did like pretty well and those that did really bad. And I had 2 genets that I lost everything and then they were no longer part of the experiment. So this is just a really good look at how I'm going to separate my genets from now on, with these being my low mortality group and this being my high mortality group. Just because we see that big gap in the center there, we want to see how different this group is from this group. But just to summarize what we've learned so far, yes, that's a bucket of corpses. I had 670 out of 2,470 centimeters square of tissue surface area died. That's a pretty significant amount. That's a pretty big chunk of coral that died during this. I had 34% of the microfragments died, which isn't terrible. 15 out of 15 of the large fragments lived. And kind of the takeaway so far is that there is some pre-existing condition of the coral that's driving mortality post microfragmentation. It's not about the size, it's something about the coral itself. This could be energy, tissue, symbionts, it could be a genetic change, it could be all kinds of things, but that's what we're going to look into. And the other takeaway is that mortality was only seen in microfragments. So next up, we want to look at the energy content. So while I was fragging everything, I took a couple of pieces that were like this big and I froze them in a freezer until I could process them. And later on, you see me here hunched over. This is why I have back pain. And I have an airbrush that's full of filtered sterilized seawater, and I am airbrushing the coral tissue off into a little bag that I'm trying to also like cinch the bag off so that nothing like sprays out. And yeah, it's super great. Before I murdered them, well, they were already frozen, so they were already dead. But before I airbrushed them, I took so many photos and I made these 3D models of them in Agisoft Metashape. Thank you so much, Sonora. And I took these models into Cloud Compare, and this was how I found a surface area of them. I wanted 3D surface areas of these to be more exact because I was doing things with the tissue biomass and things. So next I took all my samples to Alabama. I put them in Robin's really nice cooler. It's my cooler now, and brought them with me. First, we freeze dried the samples we lyophilized them is the word, to get as much water out as possible, or all the water out. They were in this for a few days. Stored them at -80°C. I dried them out. I separated them. I crushed them, homogenized them. The big takeaway is that I did ash-free dry weight for tissue biomass. That's just a process where I take some of that freeze-dried coral tissue and I know the weight that it came from. I know the weight that it is. I put it in a crucible and I bake it overnight at a really easy 500°C for about 12 hours. And then I weighed next day what was left over, which is just ash. So that's any non-organic materials left behind. So then the difference between those weights is all my organic materials, right? It's all my tissue, all my carbon, all that kind of stuff. I can then know the weight of biomass and normalize it to the surface area it initially came from to find the tissue biomass per centimeter squared of tissue. So that's why I did those 3D models. Used some of the same dried coral tissue for proteins and carbs. I extracted them and I used a couple of different assay methods. I'm not going to go super in depth into it. This is one of these assays. But for example, I got the proteins into solution, added an indicator dye that turned them a different color based off of how many proteins there were, read that color with a spectrophotometer, that's the word, and calculated protein amount in energy based on a standard of known concentrations. If you want to ask me more about that, we can talk about it later. Let's not get too much into it. So right here we have tissue, right? So, and then we have our proteins and our carbs. And then on the bottom there is our mortality rates, our high mortality, and then our low mortality groups. Weirdly enough, the high mortality group had an average higher carb amount. But the main takeaway from all 3 of these is that I ran some Welch's t-tests on them and none were significantly different. We have all of these points all the way up here. The data is very spread out, meaning that there's no glaring difference between the high and low mortality groups in either of these 3 physiological metrics, which is super interesting and not at all what we thought we were going to see. So the 3 of them probably do not drive survivorship post-microfragmentation, right? Because we didn't see any big difference between those that completely died off and those that all survived. This is totally unlike thermal stress and bleaching, which is kind of a game of energy reserve maintenance on a longer term. So surviving microfragmentation immediately is not a matter of energy reserves, most likely. So lastly, we'll look at the symbiont community composition. There's me again with that coral homogenate, that cup of goo. So I sampled from that. I preserved it in ethanol. Again, brought it to Alabama, extracted the DNA, and I ran real-time qPCR. We are not going to go into it too deeply. But that's me doing it. It's a whole lot of pipetting. But the idea is I run 1 plate for each of the 4 Symbiodiniaceae genera. I run a Symbiodinium plate and a Breviolum plate and a Phytocopium plate, so it's a whole lot of pipetting. But what that is going to give me is I run the samples against known concentrations of symbiont genera, kind of like those protein assays. And I can estimate the amount of that genera of algae are contained in the sample. And I can take those numbers and find the proportion of each symbiont genera in a sample. Oh yeah, there's the PCR machine. She's a beaut. So it's not supposed to look like that. So first I wanted to kind of zoom in a little bit. These are our 4 high mortality guys. These 2 had 100% mortality. These 2 were like 70% mortality. There's kind of some differences between the 4. We were really thinking that we would see them like all be full of duristinium and we'd be like, oh my God, it's trenchite. But there's nothing glaringly unified about the 4 of them. So it's most likely that symbiont community doesn't drive mortality post-fragmentation, but this is a pretty small sample. So just keep that in mind. So next we wanted to look at, right, if after a long time in the nursery, if the microfragments and the large fragments would shuffle to duristinium due to the stress. So first we'll look at pre-fragmentation. These are some of our guys. I know it's a little bit of a lot. These are the microfrags on the bottom excuse me. On the bottom is the microfragments in May. So it's kind of a lot to look at. So we'll break it down a little bit. Mainly what we were thinking about is not that. Dang it. Can I do it over here? I can do it over here. Mainly we kind of wanted to look at that durastinium, right? So I ran a permanova to look for statistical differences between these two groups, and there were significant differences, likely because down here in May in the microfragments, we see these kind of have a lot more cladocopium than they used to, but it's not the big change in durastinium that we expected to see, right? We expected to see them fill up with durastinium in May. But we didn't super see that change. I am just struggling with these slides. These are those same ones that we looked at before the pre-fragmentation. Now looking at the large fragments in May, we see something very similar. So there is still from the permanova a significant difference between these two groups. But again, specifically looking at the durastinium, we see kind of the same thing that we saw in the microfragments, right? Except for this one over here, who just, we don't super know what he was doing. But again, a statistical difference, but not the big change we were expecting to see. Lastly, just to show them side by side, here's the microfragments in May and the large fragments in May. So we already just saw these. The gaps are from DNA samples that really unfortunately didn't extract. Don't ask me about it, it's a sore spot. Comparing these two groups yields no significant difference. So that is what we expected, is that the mechanical stress of microfragmentation didn't cause any symbiont shuffling. Though we do see some really interesting differences in some of these samples, especially this one here, and a couple in here, we see some pretty big differences. So there's not an overall difference in shuffling, but there's a couple examples here and there of different shuffling, which hasn't been talked about or looked at before. So it'll be interesting for future studies. So just to summarize real quick, the symbiont community did not drive post-microfragmentation mortality. The opportunism in the study I talked about before was most likely due to husbandry. Those corals were in the nursery for months to years, which is probably why they eventually saw that change. The water tables were shaded pretty heavily, and there were some other factors at play there. And there was no immediate opportunism following microfragmentation. So no shuffling due to the mechanical stress of cutting them, but there were some differences. So it does need to be looked at. Perhaps in something a little bit longer term, they can look at if the microfragments, are eventually different, not from mechanical stress, but maybe from the stress of being really small little guys. I certainly feel that. So just to wrap up, kind of the big takeaways so far were that microfragmentation causes significant mortality of coral tissue. Regardless of the physiological characteristics of the coral, the large fragments always survived, and most of the microfragments, most of the genets saw some of the microfragments die. So, but looking at the physiological characteristics that we've looked at so far, surviving fragmentation stress is probably not a matter of energy reserve maintenance. There's something else going on in the coral that I unfortunately don't know what it is. And the mechanical stress of fragmentation does not cause immediate symbiont shuffling within 2 months, but maybe in the future down the line. We will be doing, I have so much more data to show you in the future. Ask me about it sometime. It's like, it'll take me another half an hour to talk about everything that we have. But I just wanted to acknowledge really quick my committee, Robin, Dusty, and Sonora. Thank you so much. Robin, this was, I mean, his project idea, and I've gotten a lot of guidance from him. I'm very appreciative of him being here today. Dusty brought me into his lab in Alabama, and they showed me the ropes on everything. I'm super appreciative of that. And Sonora has just had so many rambling meetings with me where I don't even really know what I'm asking from her stats wise, but I know I have questions. I did many a collection dive because there were multiple iterations of this experiment, but Chamoy came out with me, Davis came out with me, Aaron also came out and with us at some point, and Robin's son River also came out and demanded more snacks from me. I want to thank Dan because he's given me a lot of advice on microfragmentation, especially when I did a really bad job the first 2 times and lost everything. And I was like, Dan, why did this happen? And he's like, you dummy. He said that to me. I want to thank Shelby and Rose. Shelby and Rose in the Kemp lab for their guidance. Shelby walked me through all the qPCR stuff. She sat with me while I did it for a week and cried every day. And she was such a big help. And Rose was their technician at the time. She has since moved on. But she's the one who showed me all the proteins and carbs, and she did a bunch of lipids with me and stuff later. That's a whole other thing. But I'm super appreciative of them bringing me on, and also Alex from Kemp Lab for his general morale support. Big thank you to Aaron and Zach, who have done lots of coral cleaning when I couldn't show up, with the toothbrush on the microfrags for a couple hours. It's real nasty. They did editing, they helped me with stats work, they did a bunch of extra reflectance measurements and lab work that weren't even in the thesis. So I'm very thankful for that. To my friends in and out of MMES for their general support and love, and my family. My funding from VI EPSCoR and NSF STEM, and also shout out to Rising Tides, who's been really wonderful to me. An additional thank you to Zach for just being everything. You're gonna be quizzed on this later, so really fast. Any questions?
Hi there. So very enjoyed your talk and some very interesting results. You mentioned it just at the end. Energy storage is often in lipids, and corals can have quite a lot of lipids. So you want to just at least give us, it sounds like maybe you looked at the lipids.
Oh, I did. I'm so glad you asked. So if you're part of the study,
I knew nothing at all. I promise. This is the first time. Full disclosure, I have done lots of lipid analysis.
She was totally a plant. Yeah, so my lipids data is going to take a little bit longer to parse through. But we did also, we did total lipids, and I additionally gave the sample some TLC, thin layer chromatography. And so we did lipid classes. Those can provide more insight, you're right, into they're kind of the big energy reserves, and the different lipid classes are metabolized differently, and some of them come from heterotrophy more and some of autotrophy. So they will tell us a lot. I could not get those results done in time because Sonora and I did look at the data and go, you know what, maybe later. Maybe I can't do this in 2 weeks. But I'm very hopeful. The sterols so far looked really interesting. Just from my preliminary look, I still wasn't seeing any major differences. But then also, my corals bleached later in the year, so I then did all of those metrics, including lipid classes, again post-bleaching. So it'll be really interesting to look at how the microfrags fared energy-wise in an actual thermal stress event.
Right, because if the lipids,
Yeah,
that could be the difference between what carries them through that
Yep.
right, more so even than the proteins and the carbohydrates, right?
Yeah.
Because that's their those are the reserves.
Mhm. Yeah.
And also, I don't think it would have mattered here, but I guess you might want to think about when the collections happen, because those lipids would go into coral reproduction, for instance. So depending on where they were in the reproductive cycle, that might.
Yeah, absolutely.
influence.
We will look at that. Okay. I'm so glad I was ready with that slide. That's awesome. I don't have like a whole extended presentation. It's just like that and one other one.
Good job.
Okay, first question, did you intentionally make those little bullet points look like gravestones?
No.
But the real question is, do you think if you had taken, so you said like when they were 50% or a bit more, you classified them as dead. Do you think if you had taken samples from those ones and analyzed it the same, that you would have found some different results? or
Yeah,
some
that
maybe
haunts me.
process?
Yeah, that haunts me. I'm not sure. It's really hard to say. I don't know how. I do wish that I had frozen them, and maybe a future study should do that, but even then, it's very hard to, I found out through like taking the tissue from my bleached microfrags, which were, spoiler alert, dying like crazy, and tried to like catch them before they totally died, and it's made some of my results maybe a little bit wonky. We've got to like parse through them because I had so little tissue on some of them. So I'm not sure how that would work. I don't know if it would be different. I tried to, across the coral, when I took those, I took 3 pieces from different parts of my like larger piece that I cut everything from. I tried to like go around and grab different pieces for the physiological metrics to try to homogenize as much as possible, but it does haunt me a little bit, but I don't think that there would be a difference. I'm not sure. That's a really good question.
Good job.
Thank you. Yeah.
First off, great presentation.
Thanks, Anne.
I know how hard you worked, those fun to see it all put together. Do you think, kind of a two-part question, one, do you think that microfragmentation is the way to continue restoration? And if not, what is it?
For those on Zoom, Sonora made a face like.
What would be an alternative, like, way to go?
Yeah, so.
How to save the corals.
Yeah.
Yeah, so I'll answer, how do we save the corals? My experience with microfragmentation as a restoration technique has been tenuous, because I am one person, and when we talk about these restoration facilities that are producing tons and tons of microfrags, right? They have like armies of unpaid volunteers and interns and students doing yeah, Zach's nodding because he's been one, doing a lot of that work, and doing it by myself it's a lot of labor, and no one really talks about the labor cost and the time cost of maintaining them. They take a lot of work to keep them clean, to keep them fed. I literally have to, that's not a joke, I clean them with a toothbrush every day for months. So, and it's hard, and a lot of that I think will come more to light later on when I get through the bleaching stuff, because spoiler alert, the microfrags bleached and some of them kind of recovered, but a lot of them started very rapidly losing tissue and they started dying off. So we want to look at that. But if our restored corals can't survive bleaching stress, then like that's kind of where we're headed, right? That's where reefs are headed, is more and more thermal stress. And then where I think we should go, I mean, my answer is always gonna be sexual reproduction, that kind of variability in genotype is super important. Outplanting monotypes, we have found through forestry and all kinds of things, is not usually like the best idea long term. And I think the symbiont work is going to be really important long term, because we do have those thermally tolerant symbionts. A lot of people think Durastidium trenchi is the answer, but there are trade-offs. So there definitely needs to be a lot more research in that area. So I don't have like the answer for you, Erin, so.
Well, to follow up on that, but your those larger fragments did have a
Mhm.
high, a good, a
Yeah.
good survival rate.
They
So,
survived really well. I'm gonna look,
so do they also, I don't know enough about this, do they also survive the fusing? I mean, do they have some of the other characteristics that are beneficial for microfrag? In other words, why
Yeah.
not use, and what it looks like you were
so
saying
like
is just
bigger, like
little
bigger
bit bigger.
frags.
Right.
Yeah, so my little bit bigger ones, even my really big ones are like, they're pretty solid, so. I'll be looking at their growth rate compared to the microfrags over time. That's the second one there. I did instantaneous calcification every month for a few months, and then during the bleaching event to see how the skeletal growth is going in those large frags and microfrags. And then I did long-term buoyant weights and also surface area measurements. So I'm gonna look at how that growth compares. Typically, those big corals, what I've like experienced data aside is that those big corals don't grow quite as fast, but you're also not seeing that mortality. So it's kind of a give or take. A lot of restorationists don't take large pieces of coral off the reef and microfrag them anymore. It depends. A lot of places take fragments of opportunity, so stuff that's broken off. But studies like these are to look at, should you take that fragment of opportunity and microfrag it, or maybe should you just like epoxy it onto the reef and leave it alone, or, you know, there's a lot of questions surrounding that.
But do the larger fragments fuse too? Does
I
anybody know?
don't think that's really been looked at, but I would assume they would. I'm not sure though.
I mean, anything that, if it's the same.
It's the same genotype,
genotype,
it probably would. But you wouldn't get that like,
You could get that same benefit of the
Yeah.
larger cones, right?
yeah. But you wouldn't get that, that like accelerated growth, which is like kind of the point, yeah, of the microfragments. Yeah.
So have you found any other ways that we might utilize to accelerate coral growth? Do you have any insights?
I'm not sure. This is also only looking at one species of slow growing coral. Microfragmentation works on plenty of like faster growing species. In my experience, it's just a lot of cleaning and a lot of luck. Cause like, man, like just talking about what I saw later on, I had a random genotype, just not bleach. I had stuff like, I cut them really small and they grew super big, and then I had some that I cut really big and they didn't grow at all. So I have no idea, Kayla. I've seen all kinds of stuff in these bad boys, but hopefully I'll be able to parse through it as we go through our mountains of data. But it's a really good question. I wish I had a better answer for it.
Just one question with your fragmentation. Did you feed your coral or did you?
Mm-hmm. Yeah, I fed them a few times a week. I think the same as you guys, because I took that advice from y'all. And the feeding did work a lot better than a previous iteration where we were like, oh, we don't need to feed them, it'll be fine. Feed them. Feed them. But they were all fed the same 3 times a week, and I did the amino acids just to really keep them happy.
I was wondering if if there's anything in the literature about this coral, like what symbionts this coral species typically uses here in the wild.
Mm-hmm.
And if we know, and or, I guess it's a 2-part question kind of, if we know, and if so, like, can you compare that to what you saw in the water tables? And is it possible there was any symbiont shuffling before you looked at what symbionts were there?
Not likely, because symbiont shuffling usually, typically what we've seen is it either takes a long time, with like the bleaching events, because you actually have the symbionts either getting kicked out or leaving. We're not quite sure how that works. And then allowing that.
You know, we just tap the screen.
Oh.
Like the physical
Oh.
screen.
No.
Keep it on.
I couldn't read that fast. Oh, man, my mind just blanked out.
So no, they could have changed
Shuffling is
before, because it happens.
No,
so
shuffling is
Okay.
slow. It's
Okay.
usually either bleaching or other kinds of like a lot of stress. The moat corals, it could have been water quality or it also might have been additional shading.
So you
Shelby,
could-
did that work?
Assume that the symbionts that you looked at are the ones that
Yeah.
were there when you collected it.
Yeah, that was the assumption. I tried to do it kind of as fast as possible without like totally freaking the corals out. So I brought them into the water table. I think I gave them about 2 weeks before I started cutting them, and then I froze those samples as soon as possible. So there were only 2 weeks, so I don't think anything in that 2 weeks would have changed them super drastically.